Well, welcome everyone to MycoTalks. I'm Joe Heitman. It's really my great and distinct pleasure to co-host this version of MycoTalks with my co-host Ilyan Ilyev today. We have two fantastic speakers for you, Michael Lorenz and Claire Hoving, who will be here speaking today. And we are deeply grateful to both the organizing committee for MycoTalks and also the MRC Center for Medical Mycology and the University of Exeter for envisioning and hosting this impactful series. Just to remind ourselves about the philosophy and origin of MycoTalks, there's a focus on diversity, including career stage, gender, geography, and different fields. We have two 30-minute talks that cover all aspects of medical mycology, followed by 30 minutes of in-depth discussion. If you could submit questions through the Q&A, that will allow us to see them. And if you could preface your question by which speaker your question is directed to, that would be very helpful. We do not always take the questions in the order that they're submitted because we're organizing the questions in a thematic fashion and sometimes combining questions when that's possible. So we'll get to as many of the questions as we can in the time available. I want to direct your attention to the upcoming MycoTalks that'll be on March 31st, featuring Dan Kaplan and Hiten Madani. And then on April 28th, featuring Kyla Ost and Gustavo Goldman. And just remind everyone that you should register for each of these sessions if you're interested in participating. I'll also note that the talks are recorded and available at the MRC website and via YouTube. So if you're not able to watch the talks in real time, you can always go back and revisit the recorded versions. I want to remind our community that there's a parallel series, the Medical Mycology Trainee Seminar Series, and we see the organizing committee featured here and their photographs. We owe them a deep debt of gratitude as well for bringing a thematic focus on students, postdocs, clinical trainees, and new faculty. Some of the recent speakers in the series are featured below on the slide here, and there's an open call for nominations for speakers for this series ahead. I want to close this introduction then by featuring an advertisement for the Gordon Research Conference on Cellular and Molecular Fungal Biology. It's being chaired by Jason Steich and Alex Brandt, and it'll be preceded by a Gordon Research Seminar for all the trainees. You can arrive a day or so early and meet each other. And the chairs for that are Abigail Labella and Charles Poorner. And these meetings are open now for submissions and abstract submissions. It's a fantastic meeting. I really encourage you to check it out. So it's my pleasure to introduce our first speaker, Mike Lorenz. Mike did his PhD training at Duke, where he worked on yeast pseudohyphal growth and the role of TOR and nutrient sensing. He was then a postdoc with Jerry Fink at the Whitehead and MIT, where he started his landmark work on transcriptional analysis of Candida interactions with host cells. He's been at the University of Texas, Houston since 2003. He's a professor in the Department of Microbiology and Molecular Genetics. And the title of his talk today is Candida-innate immune interactions, surprising lessons from comparative transcriptomics. So welcome, Mike. I'm gonna stop sharing and hand over the platform to you.
Thank you, Joe. I appreciate that kind introduction. Let me get myself into presentation mode and find a pointer. At the beginning, I would like to thank, again, the MRC Center in Exeter for arranging these very engaging seminars. Especially Jules Bristow, who is the brains behind the operation and really makes these presentations work. So what I'm going to do today is tell you some lessons that we've learned from our comparative transcriptomic efforts over the last few years that really point to metabolism as being a very important part of the response of fungal pathogens, in our case, mostly to Candida species.
I'm sorry, Mike. Yeah. Sorry, Mike, you're in the presenter view at the moment. If you could go up to swap displays.
Okay. I
There
have
we go.
multiple screens. I have multiple screens in front of me, and I never quite know which
Looks
one we're
perfect
seeing.
now.
So there we go. Okay.
Thank you.
So as I said, what I'm going to try to do is to tell you a little bit about our lessons about how fungal pathogens have adapted their central metabolism to take advantage of nutrients they find in the host, and how they've used these pathways also to improve their own fitness in terms of affecting innate immune function in these cells. And in this sense, I was delighted to be able to follow Al Brown's talk last month, because Al's group and my own have had similar ways of thinking about these things for the last few years. So this is a slide I probably don't need to include in this talk, but it's from a paper written by a number of our colleagues, including Gordon Brown and Stu Levitz and Neil Gow and others, about 10 years ago now, that really highlights the problem of fungal pathogens. And it does so by putting some pretty stark numbers on these diseases. I use this slide a lot when I'm talking to non-fungal audiences, and to be honest, it often elicits a gasp from bacterial pathogens, from people who do bacterial pathogenesis, who really don't consider the problem of fungal pathogens very often. Now, these numbers are obviously quite staggering, and the mortality associated with them is really shockingly high for many of these organisms. But I also want to highlight on this slide another issue, and that is that most of us are exposed to these organisms almost ubiquitously. Sorry, that's not where I wanted to go. Almost ubiquitously. So Aspergillus fumigatus, for instance, is a very common airborne pathogen. We breathe it in all the time, every day. Candida albicans is pretty close to an obligate mammalian commensal. We see Cryptococcus quite frequently as well as many of these other. The second talk today will be on Pneumocystis, which is a basically obligate human commensal. So why are we, most of us at least, protected from these infections, whereas immunocompromised and otherwise debilitated patients suffer what can be really life-threatening and fatal infections? And in large measure, at least for Candida, but for many of these others as well, it's the innate immune system that protects us from these interactions. So we have used for a number of years a model system that basically feeds Candida species to mammalian macrophages, and then asks what the outcome of that co-culture is. And I'll show you a movie of this in a moment. But we use macrophages for a couple of reasons. The first is that they're quite easy to propagate in the lab. They're easy to obtain from mice when we want to do primary cells. But the outcome, as you'll see, is that Candida wins this interaction almost all the time. That gives us experimental space, a range of outcomes that allow us to identify Candida mutants that are less virulent, either in macrophages or in whole animals. So we like this model. And I have to say that 25 years ago now, which seems like a long time to me, I sat in Jerry Fink's office where he showed me this quite grainy black and white movie where you can see Candida inside the macrophage here beginning to germinate. And at the end, the hyphae gets longer and longer and eventually ruptures the macrophage, as you can see right about here. So this was taken about 1995 with the best technology that was around then. More recently, we have taken this movie, which I'll start using an imaging plate reader, and you can see technology has come a long way in this. These are propydium iodide stained macrophages, so every macrophage that turns red is one that has been killed by the Candida cell. This, you can see, is relatively quick. You can start seeing macrophage death within 2 hours. And obviously the avid hyphal growth that really takes over this entire co-culture. I'll move to this beautiful still picture, which was taken by Pedro Miramon, a senior scientist in my lab. And you can see the red stained Candida. Green is the actin of the macrophage, and blue is the macrophage nucleus. And obviously, even in this still image, you can see the dynamism of this with multiply infected macrophages, Candida forming hyphae, and in some cases here being phagocytosed. So we use this model as a platform for gene identification. And to do so, we initially used microarray technology using homemade glass slide microarrays that were relatively crude and had a relatively limited dynamic range. Obviously, more recently, we've turned to RNA sequencing as a way to do transcriptomics, and that's mostly what I'm going to talk to you about today. So we asked a simple question: what is the gene expression profile in this cell, which is inside a macrophage, relative to this cell, which is not inside a macrophage? So what are the genes that are induced after phagocytosis? And we have recognized for a number of years now that the predominant recognizable response of Candida is a profound nutrient starvation. So on this right side of this pie chart are categories of genes that are all involved in carbon metabolism in some form or fashion. I'm going to come back to the yellow pie section a little bit at the end. These are genes of unknown function, and we are turning our attention to those genes now. But what does Candida induce inside the macrophage? Well, it induces genes involved in the import of amino acids or peptides or N-acetylglucosamine or carboxylic acids, all nutrients that Candida can survive on as a sole source of carbon. It also induces the beta oxidation of fatty acids. It induces gluconeogenesis in the glyoxylate cycle. It induces intracellular transport of intermediates in these metabolic pathways. So really, this is fully half of the response, is simply remodeling metabolism to account for the nutrients it finds in its new environment, which is the phagolysosome. So is any of this important? And in fact, it is important. We've knocked out some of both gluconeogenesis and the glyoxylate cycle. And you see this is the standard tail vein model of disseminated candidiasis. And mice survive substantially longer when infected with these mutants than with a wild type strain. At the same time, Al Brown's group was doing some of the same experiments. And so this on the left is our data. This on the right is data from his group. You can see basically the same finding. ICL is also attenuated. This is another gene, PCK1, that is dedicated to gluconeogenesis. And then for the sake of completion, this is a gene dedicated to PYK1, pyruvate kinase, dedicated to glycolysis. So it appears from this data that Candida in the animal needs to use both glucose, but in some niches also needs to use alternative carbon sources that have to be metabolized through the glyoxylate cycle and gluconeogenesis. Now, we started with perhaps a naive thought, which was there might be a predominant nutrient that Candida is finding in this environment. And because many of these pathways don't exist in mammals, that predominant nutrient might have metabolic pathways that are amenable to being, or they might be druggable. So we started thinking about what are the nutrients from our genomic experiment? What are the nutrients that we see that are induced or present in this environment? And we hit upon three of them that we really wanted to study in greater detail. That was amino acids, carboxylic acids, and N-acetylglucosamine, all of which should be fairly prevalent in a number of host niches. So we used, again, mutations to knock out the ability of Candida to import amino acids from the extracellular environment, to import carboxylic acids from the environment, or to metabolize N-acetylglucosamine. And as you see in this tail vein injection model, the phenotypes are modest to non-existent. And that was sort of a disappointment to us for a while, because these all had great in vitro phenotypes. They were very strong. But when we went into the animal, now they didn't really affect virulence very much at all individually. CRISPR has made experiments possible in Candida that were not really feasible before. And so we said, well, maybe it's not one thing they're eating, it's all of them. And so we knocked out all three pathways in a single Candida strain. And now you can see a virulence phenotype like the gluconeogenesis phenotype I showed you on the last slide. So we actually do think Candida is making use of multiple carbon sources simultaneously in different host niches here. You can see this in an ex vivo model. This is macrophages, and we're tracking propidium iodide staining, as in the movie I showed you a minute ago. And as you can see, if you do this with a wild type Candida strain, the macrophages start dying a few hours into this experiment and get to a point where most of them have been lysed by 10 or 12 hours into this. Beautiful work by Anna Traven has suggested there's a bimodal mechanism for this, one of which is caused by Candida, and one of which is caused by glucose exhaustion in the media. But when we do that with a mutant that lacks all three of these metabolic pathways, now we see very little, if any, death of the macrophages here. This bimodal mechanism that the Traven lab has described is not seen here because these cells are also trapped within the macrophage and do not have access to the glucose and so cannot exhaust it. So we knew from this work that Candida uses alternative carbon sources like amino acids and like lactate and like glicnac as a source of energy during infection. So we next asked the question whether these were just nutrients. Is this just a source of biomass, just a source of energy? And our first hint of this was really an experiment that was done by accident in media that we had made improperly, I have to admit. We started using a media that used case amino acids as the primary nutrient as we were trying to study this phenomenon. And as you can see here, Candida grows extraordinarily well, almost as well as it does on glucose, when case amino acids are the only source of carbon present in the media. We like studying things that are acidic because the phagolysosome was supposed to be acidic. So we adjusted this media to a pH of 4. As you can see here, that's a little bit below what a mature phagolysosome ought to be. When we did this, we also buffered the media. And one of my students, Aaron Carmen, one day claimed he did this on purpose, but we think he probably just forgot. He didn't buffer the media. And you can see what happens here, which is a dramatic 3.5 unit rise in the pH of this media in a relatively short time frame. Erin was about done, and we turned this over to Slovena Vilkova, who's now runs her own group in Jena. And Slovena did a lot of experiments that I'm not going to show you, but I'm going to summarize in her model that she put together here. And what we think is happening, at least in vitro, is that Candida recognizes extracellular sources of amino acids through a system that's conserved in many fungi. It's been worked on a lot by Per Lungdahl's group, called the SPS system. This activates a transcription factor, STP2, that increases the uptake capacity for these amino acids through a dedicated amino acid permeases. Once in the cell, we want this part of the amino acid, which is the carbon backbone of the amino acid. This nitrogen group then is somewhat in excess. This is deaminated and essentially becomes ammonia. And through a clever set of experiments Slovena did, she was able to demonstrate that under these conditions, Candida actually off-gasses gaseous ammonia. Ammonia is obviously a strong base, and that is what drives the pH upward. Now, while we were doing this, we were running some control experiments, and in fact, sort of fortuitously came at another set of phenomenon that are related. And that is that amino acids are not the only nutrient that can do this. Carboxylic acids also do that. You can see here amino acids in the pink line, but lactate and alpha-ketoglutarate in blue or in red and blue respectively. And the pH goes up almost as much here. In acetylglucosamine, an observation first made by Jamie Konopka's lab, also leads to a significant rise in the pH. So this comes, the pH rise in glicnac comes also with the excretion of ammonia, but with carboxylic acids, it does not here. So does this have impacts or ex vivo? And the answer is it does. So this is, again, our mutant that knocks out all three of those pathways. And you can see, in contrast to these lovely hyphae forming inside the macrophage here, these cells really don't germinate. And so we know that one inducing factor for germination in Candida is neutral pH. And so we asked the question, which really wasn't appreciated at this time, of whether Candida is actually in an acidic phagolysosome or not. And in fact, it is not. We used lysotracker red, which is an acidophilic dye that accumulates in an acidic phagolysosome. And you can see by quantitating the fluorescence here, a wild type cell is really in a neutralized phagolysosome. When we start knocking out these various pathways, they're additive. And once you get to one that lacks all three, now these cells are really in an acidic phagolysosome. So we took all of this data and hypothesized that Candida is using these basic metabolic pathways to neutralize the phagolysosome, induce its own hyphal morphogenesis, and then escape the macrophage. The nutrients that we identified here, our genomic evidence suggests that the phagolysosome are rich in these nutrients, and there is some additional data to suggest amino acids and lactate, at least, are probably abundant in this environment. Mutants that are unable to metabolize these nutrients then are impaired in a number of aspects of host interaction, up to and including virulence in a whole animal model. Now, since we first published this model, there's been a couple of papers that have come out that challenge aspects of it, including one from Sergio Grinstein's group and Johannes Westman that suggested that the alkalinization capacity of Candida is not sufficient to overcome the power of the macrophage vacuolar ATPase. That is to say that the macrophage can acidify the phagolysosome faster than Candida can neutralize it. They also suggested that germination begins in an acidic phagosome, not a neutral one that as we had described here. And that neutralization, which does happen, actually results from membrane damage caused by the hyphae. Subsequent work from Per Lundahl's group suggested that generation of ammonia by glutamate dehydrogenase was not actually required for macrophage escape. So these are conflicting pieces of data. And I can't answer the question now, but we are actively thinking about this and actively working on it. A talented PhD student in my lab, Hannah Wilson, has been working on this, and she thinks that none of us are entirely correct with this model, and none of us are entirely wrong with it. And she has a model that she's testing now to reconcile all of these models, all of this data. And if I ever get asked back, I hope I can describe this to you at some point in the near future. Okay, so the second bit that caused us to ask whether these nutrients might be more than just an energy source was beautiful work done by Jules Inne when she was in Al Brown's group. Now, this is actually data from my lab, but it nicely recapitulates something that she published a couple of years ago. And that is that the presence of lactate increases the resistance of Candida to a variety of stresses, here shown calcul-4 white and Congo red, which are two cell wall stress agents. And this acquired stress resistance from the presence of lactate is really quite striking. But we repeated this experiment and also used our amino acid media, and you can see there is some acquired resistance to that as well. So we went on, and so we took from this that the absence of glucose, or perhaps the presence of certain alternative carbon sources, was going to lead to a pattern of stress resistance, really irrespective of what that alternative nutrient was. And so that's the attitude we took into this experiment. And it turned out that that was not correct. So cell wall stresses, lactate really protects against that. Reactive oxygen species and reactive nitrogen species, amino acids are very protective against those insults. Whereas all of these alternative nutrients increase the resistance to antifungal drugs. And in this case, lactate also increases resistance to osmotic stress. So what we saw here is a unique pattern of stress resistance induced by these different carbon compounds. And it suggested to us that perhaps Candida is using the nutritional makeup of whatever host niche it is to anticipate the stresses most likely to occur in that niche. So this is another way that Candida has perhaps adapted its metabolism to serve its primary lifestyle as an opportunistic pathogen. Okay, so What I've shown you is Candida uses a lot of nutrient sources in vitro. We sometimes refer to this as a smorgasbord or an all-you-can-eat buffet of this. And it seems to do this largely in parallel, so it's using multiple things at the same time. Assimilation is of these alternative carbon sources is a source of energy and biomass, of course, but it also generates products that may impair phagocyte function and provide signals to the cell that is in particular host environments. So I want to go on now. This is the pie chart I showed you before. This is the left half of this, again, are genes that are uncharacterized. And as we have been pursuing our metabolic work, we've always been interested in what these genes are. Now, I say these genes are uncharacterized. And what I mean by that is that they have no ascribed function in Candida, but they also are not homologous to genes with ascribed functions in model species or other fungi. So we really know very little about this set of genes, which is about 100 genes or so. We initially wanted to take an approach of 100 genes was too many to study to begin with, so we wanted to narrow this down. Today with CRISPR, we're actually almost done creating a library of all of these mutants, which we're going to put through a variety of host-related conditions. But to start this off, we decided we could narrow this down using a comparative transcriptomic approach. And we started that by just looking at different Candida species. And this was done by a person who was at the time a very, very talented undergraduate student from a neighboring university across the street, Rice University. Shelby is now in Joe's lab, or just about to defend her PhD thesis there. But you can see here, this is Candida albicans with macrophages and a variety of these other species, some of which make hyphae or pseudo-hyphae here, and others do not. We know that Candida albicans, at least in experimental animals, and Candida tropicalis are quite pathogenic. Clinically, Candida parapsilosis is fairly prevalent, but some of these other species are rarely or almost never seen in a clinical setting, and they're not virulent in experimental animals, and they are unable to affect macrophage killing under these conditions. So we thought we could use this as a tool to identify what the responses and the genes were in Candida albicans that were perhaps the most important. To do that, we used comparative transcriptomics, which was analyzed by Andrew Pountane, who was a postdoc in my lab and is now at NYU. And what you see here is principal component analysis, where these three species, albicans, dubliniensis, and tropicalis, all clustered together. They are the most closely related to each other, and with the exception of dubliniensis, the most virulent species. These are related species, parapsilosis and Longosporus, and then up here by itself is Candida lusitania, which happens to be the only haploid species in this analysis. In all of them, what you see is phagocytosed cells cause a rightward shift in the principal component analysis, and as I'm going to tell you, this really has to do with carbon starvation. Andrew went on to use k-means clustering to identify patterns of gene expression. And what we were really looking for is what's seen here in cluster 2, which are genes whose fold induction in the macrophage is higher in the virulent species than the less virulent species. I'll point out right here, this is a cluster of genes that are specifically induced in Candida parapsilosis, and I'll show you just briefly at the end a little bit about that. So what's in this cluster? It turns out that in this cluster, there are 90-something genes that are induced in all of these Candida species here. What those 90 genes are is shown here in GO-term analysis, and it is all about carbon metabolism. So the common response that's conserved between these pathogens is all carbon metabolism. And in fact, Aaron Mitchell's group published a transcriptomic approach with mutants lacking the glucose repression factors MIG1 and MIG2. And you can see here that over 70% of this common response is governed by the MIG glucose repression system. Andrew delved into this data in a little more detail and came up with something that we found really surprising. We've used genes like ICO1, which is part of the glyoxylate cycle. Gen2 is a carboxylic acid transporter. PCK1 is part of gluconeogenesis. We've used these as as sort of sentinel genes for examining what's happening in Candida cells. This is now not fold induction, but it's absolute expression levels, the total normalized read counts in each of these experiments. And you can see they all show up at basically the same level. So the reason that Candida albicans ICL1 is so highly expressed is not because it's expressed or highly induced, is not because it's more highly expressed. It's because its expression in the control condition is lower than some of these other species. That holds true with all three of these genes, whereas the fold induction is driven not by the expression in our phagocytosis condition, but our control condition. Andrew then went to look at this in more detail, looking at all of these genes. And what you see here is the fold induction on the y-axis and the absolute expression on the x-axis. And this is phagocytosis cells. And you can see there is no correlation at all between how much a gene is induced and how highly it is expressed in the condition we care about. Instead, it's exactly the opposite. There is a very strong negative correlation between fold expression and the expression in our control cells grown in media alone. So we've been studying for years, based on fold expression, a set of genes that are induced very highly. And we thought that that was macrophage driven. And what it really is, we've been studying glucose repression all this time. That was a bit of a sobering thought, but it led us to try to reanalyze some of this data in a different way. And that is to look for genes whose expression in the virulent species really was higher than it was in the less pathogenic ones. You can see a graph that shows that here. You will recognize a few of the genes on this list, TEC1, BRG1. And in fact, there are 26 genes that are expressed at an absolute level higher in albicans than in the other species. Those are shown here in this heat map. You can see the high expression. You can also see that control cells and phagocytosis cells are not that much different for most of these. The orange squares on the right here are all genes that have been associated with hyphal growth or biofilm formation in some manner. Now, this is not terribly surprising. Hyphal growth in albicans is particularly important for its pathogenesis, but this really indicated that what Candida albicans thinks is most important is this ability to form hyphae. You will note in here there are a couple of uncharacterized genes in this pathway. We're now looking at those as well. This gene in particular is the Candida albicans homologue of Dig2, which is a filamentous growth regulator in Saccharomyces cerevisiae. It was annotated in a haploid transposon screen done in Judy Berman's lab that this gene might be essential. It's certainly not essential in Saccharomyces, but our initial experiments suggest that it is indeed essential in Candida albicans. I mentioned Candida parapsilosis. This graph, bar chart, shows you the number of genes in the genome as a whole that are induced by macrophage phagocytosis, and the number of genes that are specific to Candida albicans that are induced by phagocytosis. There's not much difference for any of these species, with the exception of Candida parapsilosis, in which more than almost 50% of the parapsilosis specific genes are induced by phagocytosis, suggesting that its evolution might be, the evolution of pathogenesis in this species might be fairly recent, which I think is consistent with Geraldine Butler's very nice work and more sophisticated work. A huge number of these are nutrient transporters. You can see here all of the Candida parapsilosis specific genes are in green, and the parapsilosis specific transporter proteins are in red. So parapsilosis has expanded gene families of nutrient transporters quite significantly. We also have turned, as have many people, to Candida auris, which I'm sure everyone realizes now is a emergent pathogen of some concern. And we've done this macrophage transcriptomics with Candida auris as well. And again, the common response between auris and albicans, and in fact the other species as well, is carbon metabolism. Candida albicans has a set of genes. About half of these genes, which are uniquely regulated in albicans, are nutrient transporters. So that's consistent with the parapsilosis data I just showed you. In Candida auris, there's a larger number of induced genes, and about half of these are also nutrient transporters, just a different set than albicans. This suggests, in fact, that one key response of these species to becoming pathogens is the expansion of gene families that enable nutrient uptake in these cells. In Candida auris, there was one other surprising finding, and that was a group of about 25 genes that are all annotated to the Go term of drug or xenobiotic transport. And again, this suggests that the multi-drug resistance that we know is as a hallmark of Candida auris, that it has tuned its own gene regulation to upregulate a number of genes, and included in the set are multi-drug resistance pumps and a variety of other things, so that it recognizes certain host stresses in order to turn on these transporters. I'll conclude with one last slide. We also did this in the gene expression profiling in macrophages, and that was quite uninformative. It did match stuff that data that Bernie Huby's group has published, but this is live Candida albicans versus UV killed Candida, and you can see there's hardly any genes. There's a very tiny, more pro-inflammatory response to dead albicans than live, but it's really not terribly significant. We compare Candida albicans to tropicalis. There's not a single gene in the macrophages differentially regulated. A couple, Candida to Parapsilosis, and with the comparison to Lusitania, there's a slightly more pro-inflammatory response to albicans than Lusitania. This might be because the cells are small, the Lusitania cells are smaller, and we were using similar MOIs here. But the macrophages themselves don't discriminate in the least between these two species. Now, finally, we were hoping for some illumination of what makes Candida albicans a good pathogen relative to the rest of the ones. And what we found was that there's a common response in terms of carbon metabolism, which means that this is necessary but not sufficient for virulence. This is largely driven by MIG-1 dependent glucose repression. There are species-specific responses, and this is where we're turning our attention right now, including hyphal and biofilm genes, newly evolved genes in some of these species, drug detoxification of Candida auris, and I've mentioned the transporter genes. So with that, I want to thank you for your attention. I particularly want to thank the people who did the work. Most of it has been done by the people shown in red on this slide. And I think we're holding questions till the very end, but I want to thank you for your attention.
Thanks very much, Mike, for a really scintillating talk. I'm going to hand it over to my co-host, Ilian, to introduce our second speaker.
Thank you, Joe. It's my pleasure to introduce Claire Hoving. She's an associate professor and Wellcome Trust intermediate fellow in public health and tropical medicine. Dr. Hoving has done her Bachelor of Science degree in the Groningen University in Netherlands, and her Master of Science and PhD degree in neglected infectious diseases and ulcerative colitis at the University of Cape Town. Later, she joined the group of Dr. Gordon Brown at Cape Town University, where she studied MTB infections. And later, when she started her laboratory, she focused on fungal infections that are affecting HIV patients, such as Pneumocystis, Cryptococcus, and Mergiomyces. So for her work, Dr. Hoving holds a number of distinctions, and I will just name a few: the NAIDS Medal from the Parasitological Society of Southern Africa, and most recently the South African Medal Research Council Silver Medal for contribution to medical science, and she's also a Wellcome Trust fellow. Claire, looking forward to your presentation.
Thank you, Ilian. Let me just share my screen. Everyone see that okay?
Yes, that's perfect.
Yes.
Thank you.
Okay, great. So thank you very much for the introduction, Ilian, and also thank you so much to the organizers for giving me the chance to present some of our work on this incredible platform. So as Ilian mentioned, I'm based in Cape Town, in the University of Cape Town, and actually our faculty is just over this hill here We're in the Institute of Infectious Disease and Molecular Medicine, and we're based in the Africa Unit for Medical Mycology Research, which was established by Gordon Brown and the MRC Center for Medical Mycology. And so we are also affiliated with the University of Exeter. So this is just an image showing you some of the Africa Unit members, and this includes the principal investigator, Rachel Gangarembisi. And the main focus of this unit is on important HIV-related fungal infections with the goal of expanding research capacity in Africa, where these diseases cause such high mortality. And we've been involved in various training programs and workshops which focus on these specific diseases held locally in Cape Town. And in Cape Town, we are also very well situated to benefit from the world-class facilities which focus on mainly HIV and TB research within our institute. We also take advantage of the clinical expertise in our institute, and that our medical school is also linked to Christian Hospital, which you can see here at the top. That's a teaching hospital, and also Red Cross Children's Hospital, which will help us translate our basic research back into patients. So today I'm going to present some of the work that we've been doing on Pneumocystis Pneumonia. And so just a little bit about the disease. It doesn't really need much introduction to this audience, but Pneumocystis Gerivacei causes pneumonia in AIDS patients, and in fact it is an AIDS-defining illness and led to the identification of HIV in the 80s. And with the rollout of antiretroviral therapy, the burdens became really low, but there's recent incidents suggesting that with the use of immunosuppressive therapy, pneumocystis is actually increasing in USA, UK, and Europe. And unfortunately, during the HIV/AIDS epidemic in South Africa, there was denialism by the government, which has caused South Africa to have the highest burden in the world. We have about almost 20% of our population are HIV positive. But this is also corresponding with one of the biggest national antiretroviral programs in the world. So despite this very high incidence of HIV, PCP is actually less commonly described than one would anticipate. And David Lowe wrote a paper in 2013 describing that this is perhaps due to attributed to diagnostic challenges in resource poor countries. There were also hypotheses based on perhaps there's a low reservoir for PCP in these populations, resulting in lower infection rates. However, we have this hypothesis that perhaps there are other pathogens that are prevalent in Africa that could influence the prevalence of pneumocystis and drive clearance in this population. So just a little bit of background about pneumocystis. So as you heard earlier, it is a very unusual fungus thought to be a protozoa, and it's highly species specific. So it will affect the human and mouse. Pneumocystis don't infect each other. And actually, now that the genome has been published, there's a lot known that it's lost a lot of its genes, and actually this has contributed to it being an obligate biotroph. So it basically needs the host to survive. So it colonizes the lung quite early on in childhood, and then takes advantage of a weakened immune system to cause pathology. And this is, of course, associated with CD4 T cell deficiency, as seen in patients with advanced HIV disease with very low CD4 counts. So just a little bit about the fungal cell wall. I work more in immunology, but just to show that now that the genome is published, you can compare. So pneumocystis has two life cycles, a cystic form and a trophic form. And if you look at the structure compared to Candida albicans, you can see that pneumocystis has lost mannans. It doesn't have any mannans. There's no chitin in pneumocystis, the cystal the troph. And quite importantly, actually, you can see that it has cholesterol in its cell wall instead of ergosterol, which is what most fungal pathogens have. And even in fact, the cholesterol, it actually scavenges from the host, so it can't make its own cholesterol. Unfortunately, this is the target for ergosterol, and so ergosterol is a target for azoles and amphotericin B, which makes treatment with these drugs very challenging. And if you look at the trophic form, you can see that it doesn't have beta-glucan, whereas the cyst still has beta-glucan. And this is, of course, the target for echinocandins. So this is where combination therapy may be an advantage, where the echinocandins can be used at a lower dose together with the antibiotics that's usually used to treat pneumocystis, where you can clear the cyst, although you won't clear the troph, but you can reduce the level of treatment. So as I mentioned, current treatment uses antibiotics, which targets the folic acid pathways, and that's the current treatment strategy for pneumocystis. So as you can see, there's quite a need to develop either better treatment strategies or host-directed therapies that can improve treatment. So as I mentioned, we had a hypothesis that perhaps there are other organisms in Africa that are driving clearance of pneumocystis, and this hypothesis actually came from a study done by my colleagues Heather Zah and Mark Nicol, who is now moved to Australia, and they established this amazing birth cohort. It's still ongoing, it's in the 7th year now, but for our data, we'll only talk about the data that was analyzed within the first 12 months. But essentially, this is a study that is trying to understand the causes of childhood pneumonia. And in children, it is globally accepted that pneumonia is the biggest cause of mortality in children under 5. It's estimated that about 6.6 million children die each year. And so in this study, they enrolled 1,000 infants, mothers and babies. They were pairs, and they took samples, nasopharyngeal samples. The children were actually recruited from 2 demographic areas in the Western Cape, and they took nasopharyngeal samples from these babies every 2 weeks for the first 12 months, after which first 12 months, and then after that, they took every 6 months, they took samples. And they looked at everything from environmental factors, infectious, nutritional factors, genetic, maternal. There was immunological risk factors. They looked at the home to see if there were people smoking in the home, and it really was an in-depth analysis. And at the time when the study was supposed to end after 5 years, there were 76,000 samples and 40,000 nasopharyngeal samples collected. Although the study is now ongoing, it's been funded to continue. So just to describe the data that was analyzed for our observations, I mentioned that infants had nasopharyngeal specimens collected every 2 weeks for the first year. And this was analyzed by Maya Lesowski, a bioinformatician working with Mark Nicol. And so essentially, once a baby had incidence of pneumonia, the 6 preceding specimens were collected and analyzed. This was matched with the baby at the same age that was considered healthy and had no pneumonia, and the samples were also analyzed as a control. Just to point out that one child could have more than one incidence in the year. And this was also age matched with the sample. And this led to 520 pneumonia samples with matching controls, and then about 3,120 preceding specimens that were also analyzed. And these samples were analyzed by 33-plex or PCR of known pathogens, and in this analysis, they also included Pneumocystis. So using this incredible data, you get an idea of what kind of organisms are present. These are in healthy control, so these would be the colonizing organisms. As you can see here in orange, RSV is actually only present if the children are ill and it's causing disease, so it's not normally present. And if you look in yellow, you can see actually Pneumocystis was present quite early on, and within 3 months, 40% of the babies already had Pneumocystis colonization, and this dropped off with time. And then looking at the incidence of pneumonia, and this is the odds ratio showing which odds certain pathogens would be present at the time of pneumonia incidence. So as you can see, RSV is very high odds of children having RSV, and this was one of the most common causes of pneumonia. So Haemophilus influenza was also associated with pneumonia, whereas if you look at Pneumocystis here, the odds of having Pneumocystis at the time of pneumonia were lower. And then just looking at some of the data that Maya analyzed. And so this is the time of event is the time of pneumonia. So this, just to point out, is not Pneumocystis mediated pneumonia. This is from other organisms. And this table represents Pneumocystis colonization and not infection. But if you have a look, so this was the time of pneumonia, and then these are the samples preceding the event that were taken. So in the controls and cases, Pneumocystis prevalence seemed to maintain the same with the odds ratio close to 1. Whereas at the time of pneumonia event, this odds ratio dropped almost half, suggesting that perhaps the organism within that's causing the pneumonia in these children is driving Pneumocystis clearance. So this led to the aims of our project. So firstly, we wanted to establish a mouse model of Pneumocystis pneumonia in Cape Town. And then we wanted to look if there were, to determine if there was a correlation of Pneumocystis clearance with exposure to specific organisms that were seen in the birth cohort. And if so, we wanted to dissect this mechanism of clearance and perhaps investigate pattern recognition receptor co-signaling as a potential mechanism. So at the time, I was just starting my research, and I visited Jay Polz's lab and to try and set up the mouse model of Pneumocystis. And so Pneumocystis can't be cultured in vitro, it doesn't survive. So it's quite a challenging model to work with. But you can passage it through mice. So essentially, we can infect immunocompromised mice, rag mice that don't have T and B cells. We store these lungs, and then we can use these in the infection model. And this shows you the disease progression. So wild-type mice generally can clear infection by 3 weeks, whereas these rag immunocompromised mice are susceptible to infection and eventually succumb. And this just shows you histology showing the brown GMS staining of Pneumocystis. So based on the observations in the birth cohort, we chose specific organisms that were either associated with Pneumocystis clearance or not. And so we have here a mouse model, which was 2 weeks. So we infected rag deficient mice with Pneumocystis. A week later, we challenged them either with Staph aureus or Haemophilus influenza, just to point out that Staph aureus is gram-positive and Haemophilus is a gram-negative organism. And interestingly, we found that Staph aureus didn't seem to influence the outcome. This is showing Pneumocystis burden RNA copies in the lung of an infected mouse. And that was comparable to a mouse receiving PBS, whereas mice treated with a gram-negative Haemophilus influenza seem to have enhanced clearance of Pneumocystis. And this was associated with cellular recruitment. And an interesting observation here is that actually the mice receiving Staph aureus also had a cellular enhanced alveolar macrophages and dendritic cells and neutrophils. However, this did not translate to helping clear Pneumocystis, whereas Haemophilus influenza seemed to drive similar kind of recruitment. However, this was potentially more specific and able to clear Pneumocystis infection. So actually digging back through past literature, this kind of observation has been seen before, and this is a really old paper from 1982, where actually rats were infected with Pneumocystis, and in the study they found this is the percentage of rats that still had Pneumocystis at the time of death, and you can see here rats that were untreated and rats treated with Staph aureus still maintained a high level of Pneumocystis, whereas rats that received Pseudomonas aeruginosa, which is also gram-negative bacteria, seem to have less Pneumocystis present at the end. And then another interesting study by Beth Garvey actually showed that heat-killed E. coli exposure in cups that were treated with Pneumocystis could actually enhance Pneumocystis clearance, and that you can see. So this is a heat-killed E. coli treated mouse, which seemed to survive better, had less Pneumocystis in the lung, compared to mice that did not receive heat-killed coli. So this suggests that there's definitely a relationship between different organisms, and one of the common points that we see in these is that they are generally gram-negative bacteria. So we looked at our mouse model again, and here we used LPS, which is the cell wall, major cell wall component of gram-negative bacteria. And in this is a survival model where we infected Pneumocystis into RAG deficient mice. We treated them for a month, after which time we stopped treating them. This was intranasal treatment twice a week with LPS, a very low dose of LPS. And we managed to significantly enhance survival of these mice. And we had to stop treatment, but I think if we carried on with treatment, we may have been able to continue with this detected response. So this suggests that perhaps using an agonist, a TLR agonist like LPS, could drive inflammatory responses that are missing against Pneumocystis and enhance clearance. So this just shows you the RNA for fungal burden in the lungs. So in the wild type mice, these fungal burdens are quite low, whereas in a RAG deficient mice, you can see that they have a higher burden of Pneumocystis. But this is reduced once treated with LPS. And this was 2 weeks post-infection. And then looking again at the cellular response, you can see similar to Pneumocystis influenza, there's a recruitment of these cells, alveolar macrophages, dendritic cells, and neutrophils, and also some cytokines. Not all the cytokines we would have expected, but definitely R1 beta significantly increased, but not TNF and interferon gamma. So just to mention, because these are RAG deficient mice, these responses are T cell independent. And this just shows you histology. You can see it's just visualizing the FACS data. This was done by FACS. You can see recruitment of cells around the tissues. So this co-signaling mechanism is not new, and it's actually been shown before with Gordon Brown's really groundbreaking study against chromoblastomycosis, where Gordon showed that, maybe just to mention, so chromoblastomycosis is caused by Phagocytosis pneumonia, and this is a lifelong fungal infection. It's a debilitating disease, and patients often need to have surgery to have lesions removed, and there's no treatment. And in Gordon's studies, they found that Phagocytosis is recognized by MYCO, but this does not drive sufficient inflammatory responses and then leads to chronic infections in patients. However, in their study, they showed that if you can add a toll agonist, they used LPS, which is a TLR4 agonist, and imiclimod, which is a TLR7 agonist. They were able to enhance these inflammatory responses, and quite remarkably, within 2 years, these lesions have been cleared in these patients. So just to point out here, I mentioned they used LPS. This is a foot pad of a mouse infected with Phagocytosis pneumonia, and by topical application of either LPS or imiclimod TLR7 agonist, this significantly reduced the fungal burden in the mouse model. So in Pneumocystis, we know that C-tablectin receptors recognize Pneumocystis. And in fact, quite recently, Andy Limper at the end of last year showed that MCL can also recognize Pneumocystis, and it's already been published that Dectin and Minkle recognize Pneumocystis and are actually associated with reducing inflammation in immunocompromised mice. And also TLR2 and 4 have been associated with recognizing Pneumocystis. So this suggests that perhaps there is a synergistic signaling mechanism with multiple receptors. So in our own data, we had these mice called PKC delta knockout mice, which PKC delta is upstream of CARD9 and is in the signaling pathway for most C-tablectin receptors. So we used these mice in our Pneumocystis model, and we found that actually there was delayed clearance of Pneumocystis in these PKC delta deficient mice. And actually a recent study by Andy Limper has shown and complemented these studies in that they used CARD9 deficient animals and found that there was also delayed clearance of Pneumocystis in CARD9 deficient animals, suggesting that there definitely is a role for C-tablectin receptors in infection. Just to point out that these mice eventually do clear infection, so they don't succumb to to disease like the rag mice do, suggesting that there are other mechanisms that can control infection. So just to summarize this part of the work, we've shown that perhaps it's a similar mechanism, that there's a defect in Pneumocystis recognition by C-type lectin receptors, and therefore the inflammatory response doesn't drive clearance. However, if you can use a toll agonist like TLR4 LPS agonist, this can enhance the inflammatory response. And this could be by other organisms driving this response and promotes clearance of Pneumocystis. So summary just for this section, the negative association, we've shown that there's a potential negative association of specific organisms and Pneumocystis colonization in humans, and that we potentially identified a key mechanism of Pneumocystis recognition and clearance in mice, and that this involves synergistic signaling between C-type lectin receptors and toll-like receptors. But whether this is a potential explanation for the lower prevalence of Pneumocystis pneumonia in sub-Saharan Africa, that still remains to be seen. And then some other work that we're interested in is identifying other organisms from this birth cohort that influence disease. I already mentioned that the babies have quite a high level of RSV infection. And of course, considering the prevalence of Mycobacterium tuberculosis in sub-Saharan Africa, it would be important to look at the relationship between TB and Pneumocystis. And then of course there are helminths. And just to mention, COVID-19 is now included in this birth cohort as the cohort has additional funding. So just to mention briefly, I don't have too much time to go into detail. So an interesting observation is in tuberculosis infection and Pneumocystis. And this is quite an old paper, 25 years ago from our institute. And these are HIV infected patients. And just to point out, so these are all male patients, and they are sorted according to demographic and race. And if you look at TB cases in these patients from a poorer area, this corresponds to lower. So if the patients have high TB burden, there's lower Pneumocystis burden, and also the other way around. If there's lower TB burden, there's also a higher chance of Pneumocystis burden. And this could suggest that perhaps there's that live TB exposure can affect Pneumocystis colonization and eventually disease. And so looking at this paper by Mark Nicol, this is not the birth cohort, but this is a study where they were looking at children that were admitted to Red Cross Children's Hospital with TB. And just looking at the association of different organisms found in these children, and in red you can see that these are defined TB, so these patients with confirmed TB versus patients that didn't have TB at the time. You can see Pneumocystis, although not significant, it is associated with the unlikely TB group, suggesting that perhaps there's a link between TB and Pneumocystis. So we did have a look. This is very preliminary data, and we've looked in our mouse model. In this case, we first gave the mice BCG, which would be a live organism treated as an infection. And these are rag mice, so they would be susceptible to BCG infection. And later, once we challenged them with Pneumocystis, we found that the BCG challenged or infected mice had lower Pneumocystis burden. And this just shows in the lung of rag mice with PBS versus BCG mice. You can see lower burden here. But I think this is a complex story because there are definitely patients with TB that have Pneumocystis. So I think the story is more complex than that. And then just very briefly, I mentioned already from the birth cohort data that the RSV levels were, the odds of having RSV at the time of pneumonia were very high. You can see like it was the top pathogen associated with pneumonia in these babies. And looking back through previous literature, RSV, which is respiratory syncytial virus, has been shown to signal through toll-like receptor 4. So looking at this data, it's a Nature Immunology paper from 2000. You can see that TLR4 deficient mice actually had higher Pneumocystis burden, and I mean RSV could persist longer in these mice than in wild-type mice, suggesting that perhaps RSV can also trigger this TLR4 pathway. So we decided to look in our mouse model as well. This is also preliminary data, where essentially we infected mice with Pneumocystis, treated them with RSV, and then looked at the fungal burdens 2 weeks later. And we found that quite the opposite, actually. Instead of what we were thinking, that perhaps Pneumocystis or RSV would help the clearance of Pneumocystis, we found that it actually exacerbated disease. And these are RAG deficient mice treated with Pneumocystis and RSV. And then these are immunocompromised RAG mice, and we also looked to see if this was the same in wild-type mice. And we found a trend that if wild-type mice had Pneumocystis, it aggravated, and RSV aggravated this infection. One thing we haven't actually done is look the other way around to see how Pneumocystis exposure affects the RSV, RSV level and pathology. And we could also go back and look at the infants that did have high levels of Pneumocystis. So there were only about 30 of the babies that developed potential Pneumocystis pneumonia, or at least higher levels of Pneumocystis. And we can go back and look and see if this correlates with RSV infection or not. So just to end off, this was actually another really old paper from 1993, which had actually shown, so PVM is a mouse version of RSV. It's a viral infection in mice. And in fact, they did show that viral infections can exacerbate Pneumocystis. And this is very similar to our study. And you can see here that in this first graph, this is Pneumocystis burden. These were the mice that had RSV as well, and the Pneumocystis burden is a lot higher compared to mice that did not have RSV exposure. They also measured the viral levels in these mice, which is something we haven't done. So how Pneumocystis actually influences viral pneumonia is not very clear. So in conclusion, to summarize, our studies support the broad notion that their co-infection by secondary microorganisms may have significant clinical relevance in the terms of diagnosis, prognosis, and therapy of the primary infection. And I think we can further exploit this animal model to determine the fundamental mechanisms of either the beneficial effect or actually the detrimental synergistic effect as well. So then I'd just like to thank the people that did the work and supported us in these studies, and at the University of Cape Town, Roanne Keaton, who supported my PhD student, Patricia Otino, who did most of these work to study, and then Mark Nickel and Heather Czar for allowing us to access their data, the birth cohort data, and of course Jay Coles and Andy Limper, who have been really phenomenal in helping me establish the Pneumocystis model and putting up with all my silly questions. And then the MRC Center, specifically Gordon Brown, they really support us down in Africa, and we really couldn't do it without their support. And then the rest of my team, and of course my funders. Ilya mentioned I'm a Wellcome Trust funded researcher, and this is the Wellcome Trust funds this project completely. So thank you, and that's the end of my talk.
Thank you, Claire, for the fantastic talk.
Great, thanks very much, Claire. We're going to go to our question and answer period now, and I will lead off with a couple of questions that have come in for Mike. And just a reminder, please enter your questions in the Q&A section at the bottom. So Mike, the first question is from Slavina Vilkova. Could the stress resistance be explained by the morphological state of the cells?
Yeah, so no, we don't think so. There are obviously differences in immune recognition by macrophages and neutrophils of hyphae versus yeast, but we've done this with non-hyphal mutant strains, and we still see increases in resistance as a result of these different nutrients.
Okay. And second question from Al Brown. prefaced by your observation that different nutrients induce different patterns of stress resistance in Canada is fascinating. To what extent does this reflect differences in metabolic flux versus differences in regulatory responses to nutrients?
Yeah, so Al, that's a fantastic question that I can't answer. But what we're doing to try to answer that question is by making mutants in cell surface sensors, like the SPS system, for instance, versus downstream components that are involved in metabolism, to understand the idea whether it's intracellular consumption of these nutrients that are leading to byproducts or signals that are turning on stress resistance, or it's the plasma membrane proximal signaling events that happen there. For amino acids and stress and oxidative stress resistance, it does not appear that the SPS system is involved in that. So we've made SPS mutants, and they also acquire resistance to peroxide, for instance, when amino acids are present. But we've not yet made the mutants, the downstream metabolic mutants, yet to confirm that.
All right, thanks very much, Mike. I'm going to hand it over to Ilian for questions for Claire.
Thanks, Joe. So question for Claire. So the purge cohort used a threshold of 10 to the 4th copies of mitochondrial LSU RNA. So how did you define infection versus colonization in your clinical cohort?
Yeah, thanks for the question. So, I mean, I didn't work on this myself, but what I understood is that there is a threshold. I'm not exactly sure. I think it was quite similar to the purge threshold, where almost all of these babies were in the colonization threshold, and there were only a handful that went over above that, and I think it was based on a copy number. Yeah, I'm not too sure exactly what that was though.
Thank you, Claire. So I'll hand it to Joe.
Okay, question from Mike from Carol Kumamoto. Fantastic talk. She's curious why lactate and glicnac have different effects on resistance to calcifluor white or caspofungin. And do you think that different carbon sources affect accessibility of the compounds to their targets?
Yeah, that's possible. Again, the short answer is we don't know. We've done some electron microscopy to look at the cell walls because Jules Inné and Al Brown had shown that lactate really causes differences in the cell wall, particularly the beta-glucan layer of the cell wall. And that makes sense because to make the beta-glucan layer, you rely on gluconeogenesis to make the glucose that goes into it. And so having a smaller beta-glucan layer makes sense. The cell walls look very similar in cells grown on lactate, on amino acids, and on glicnac for that. So we don't think it's gross differences, but that doesn't rule out that there could be subtle differences in wall composition that might affect permeability of the cells to any of these stress agents. I will say that your second comment about whether these compounds or their metabolites might be affecting the sensitivity, for instance, to these agents is a good one. We've recently done some experiments to suggest that a small part, certainly not all of it, but a small part of the induced resistance to peroxide of cells grown on amino acid is that the peroxide oxidizes some of the amino acids. That represents about 30% of the induced resistance of just reducing the effective concentration. We've not done those kind of experiments yet for calcifluor white, Congo red, or the other nutrients, but those are good experiments to pursue.
Great. I'll pass it back over to Ilian.
So Claire, there is a question on the bird cohort, which has been followed very well. Is the comparatively to other regions overall lower incidence of pneumocystis in Africa seen in this group of children as well? So this related more to if there is actually some artifact of the low diagnostic capacities in different regions in Africa.
Yes, I think I forgot to mention, so the birth cohort is HIV negative children. So they're actually HIV positive children are excluded. So this wouldn't be HIV-associated pneumocystis. And also there are more studies coming up now. My colleague Sean Busselman published a study saying that there is perhaps more pneumocystis around, it's just not being detected. And I think epidemiology data is just not available. So it could be that there is still this high pneumocystis burden that's just not being detected. Although there are still other studies, like in Uganda, I think there was a cohort where the patients were tested and there just was lower pneumocystis. So I think there's from both sides. But for this cohort, they are HIV negative children. So we're looking more at colonization and not Pneumocystis pneumonia.
Thank you.
Okay, Mike, a question from Julia Kohler. Do you know how Candida alkalinizes the medium when growing in lactate?
Not entirely. There are a couple of possibilities. The first is we know that growing on glucose acidifies the medium. And it does so because glucose produces a lot of protons. When you're running on gluconeogenesis, you consume those protons. And so that's one mechanism probably by which the pH goes up. But these carboxylic acids like lactate are also acids and they drive the pH down quite a bit. So as Candida consumes those as carbon sources, we think it's basically trading off energy for the ability to neutralize the medium. I also wanted to say, I showed a grainy, the old black and white movie, and Julia corrected me in the chat and said she's actually the one who took that movie, not the other people who were credited with it. I've heard a couple of different stories. I used the one that I put up there because of that's what I got from Jerry, so I'm going to blame it on Jerry Fink. But let me give credit to where it's due, and that was Julia's product.
Great. There was a second part of Julia's question. It has to do with the incidence of certain Candida species in pediatric bloodstream infections that are at her hospital. And apparently Candida lusitania is the third most frequently isolated, and Candida parapsilosis is even more common than that. And then the Ritter question then is, does that imply that the metabolic hypothesis of virulence may not be completely generalizable?
We described that the conclusion of that is that the metabolic adaptations that we observe are necessary but not sufficient. We see the same kind of metabolic adaptations in species that are almost never seen clinically. We excluded some of this data from some of our analysis because they were really far enough away that it screwed up the rest of the conclusions we could make. But we see the metabolic adaptation in Debaromyces hansenii. We see some relic of it in Saccharomyces cerevisiae even. So I really think that the metabolic adaptation comes from glucose derepression led by the MEG system. And that each of these species, Lusitania, parapsilosis, albicans, have unique adaptations as they've evolved some degree of pathogenesis for that. And like I said, for parapsilosis, that was the induction of a lot of transporter proteins that are linked to metabolism, of course, but they transport a lot of things beyond just simple nutrients.
All right, thanks very much. Back over to Ilya.
So Claire, it's more a question about this interesting data that you have and that historically have been collected. So RSV infections actually exacerbates the outcome of pneumocystis infection. And then you have the staph infection, which is actually having the opposite effect. So is that due to the immune response that those two types of infections and viruses versus specific bacteria are inducing, or is it because of the type of disease? What are your thoughts about that?
Yes, I think we included the RSV thinking there could be this similar mechanism involved, which I think it's not. It's probably using a different mechanism. And so for the bacterial infection, I think it warrants this co-signaling mechanism, whereas with RSV, I don't think so. And in that paper, they suggested, although they don't really know the mechanism, that perhaps it's just the host is busy responding to one infection in the viral infection and therefore doesn't respond to Pneumocystis or causes exacerbated disease. So there's definitely two, I mean, I think there would be two different projects. And I mean, we also have some work on helminths, which originally we also thought, you know, some data linking the fact that helminths can coat themselves with gram-negative bacteria. And in our models, helminths can also enhance this kind of clearance. So whether they're all linked, I mean, they're all quite different diseases and organisms. And, you know, we were looking for a common trend, but I'm pretty sure there's quite a lot more involved. I mean, co-infections are quite difficult because they would obviously initiate different immune responses in very different ways. I don't know if that's answered the question.
No, that's great. It's a difficult, difficult directions to follow, but great that you have a mouse model where partially some of those can be investigated. Thank you.
Okay, a question for Mike from Bernie Huba. Could Candida albicans hyphal formation in macrophages have already been initiated before the full metabolic adaptation to the phagosome, and the hyphal form is optimized for growth in the phagosome environment?
Yeah, that's an interesting question. It's one we've been considering for a while. Do we just dump Candida into this highly hyphal-inducing media that contains serum and is a neutral pH, and it initiates the transcriptional program early enough where we're not seeing germination until they're inside the macrophage, but it's sort of the cell's already going down that path? We don't, well, I'll say that could explain some of it. We don't think it explains it all. Recently, Hannah, a student in my lab, has started pre-incubating cells in Hank's balanced salt solution, HBSS. That does not induce hyphae in Candida. So the way she does this experiment is to put the macrophages and Candida together in HBSS, let them go for 30 minutes. We see little to no germination of Candida during that time. And then she washes off the extracellular Candida cells and replaces that with DMEM or RPMI. And the cells, the Candida cells still germinate inside the macrophage, and they do so with a similar, at least kinetics, to just keeping them in hyphal-inducing media the whole time. I am not sure whether the yeast or the hyphal cells are really better adapted to that. I can say, and I'm sure, you know, as Bernie knows as well, that non-filamentous Candida albicans cells survive pretty well in macrophages for a pretty long time. And we can't really directly compare that to the hyphal cells because they escape the macrophages within a couple of hours.
Great, over to Ilya.
So your data in rack knockout mice is showing that adaptive immunity plays a very important role for protection against Pneumocystis infection. And work from Jay Kosk has shown that natural IgM antibodies are very important for the initiation of the innate immune response and for the protection. So if you think along those lines, do you think like the adaptive immunity, that's very general, but do you think that lack of adaptive immunity basically is propitiating the response to Pneumocystis, or it's actually driving innate immune response that then is leading to inflammation, and then because of that you get the susceptibility and the death?
Sorry, could you repeat the question? So do you think that our model is driving adaptive immune responses that are clearing the infection?
Yeah, so is, you know, have you looked if it's basically a clearance problem, or is it a problem because of the lack of clearance? There is increased inflammation and lack of adaptive immunity, which can suppress that, is actually contributing to that death that you're observing in mice lacking adaptive immunity.
Yeah, so we thought about that because LPS would drive this massive inflammatory response, and the dose of LPS we use is really low. And it seems that the route of administration is also quite important because we did IV LPS and we didn't get the same clearance. So it seems to be you need a very localized response to drive us this clearance. And I think that down the line with adaptive immune responses, you know, if it's tailored in the innate immune response mechanism, if it's tailored in the right way, it will target Pneumocystis directly, because LPS could drive this massive big cytokine storm and inflammatory storm. And in fact, I'm just thinking in HIV patients, they have also leaky bowel where LPS can escape and go into the blood, but that doesn't translate to enhanced clearance. So I think it really is a localized response within the lung.
Okay, thank you.
All right, a question, Mike, from Matt Anderson. And he's noting that N-acetylglucosamine is frequently encountered by Candida in the gut as a result of mucin metabolism. And his question is, have you tested induction of stress resistance when grown on mucin or with a mucin degrading bacteria nearby?
No, we haven't. That's a great suggestion. We actually use a mucin-containing media in some of our work to mimic saliva in oral biofilms, and Candida does okay in that media, so it can break down mucin as well. But we've not tested that. We would predict that it would look like Lignac, but we haven't obviously done the experiments yet.
Okay, maybe I can jump in with a question for Claire. And if you mentioned this, I may have missed it, but is there an interaction between COVID and Pneumocystis?
Yes, I mean, that's an interesting question. And we did have a study right in the beginning with COVID that we were going to look at this co-infection, because my colleague in the clinics was saying he suspects there would be quite a lot of Pneumocystis in COVID patients because it looks like they're presenting with Pneumocystis, especially in the patients that were in ICU, which had a lower CD4 count. But it's not something we looked at in the end, and I'm not familiar with any of the studies looking at Pneumocystis specifically with COVID infection. So yeah, I don't know of any studies that looked at co-infection.
Mm-hmm. Maybe room for future investigation given the links with Aspergillus and Rhizopus that have been made. Okay, there's another question that came in from Mike from Mirat Kalem. Do different carbon sources impact protein synthesis globally in Candida to cause stress resistance or sensitivity? And if so, does it differ for different Candida species?
Yeah, I didn't mention this, but yes, the carbon sources do affect protein synthesis. We talk mostly about the induced genes, but a common response amongst genes that are repressed by phagocytosis is the downregulation of ribosomal protein transcription and things like translation elongation and initiation factors. So yeah, there's evidence that globally translation slows down. How this might impact the stress resistance, we don't know. In Saccharomyces, in stress conditions, they also turn down ribosomal protein synthesis. But there are some systems to ensure that there are priorities or that certain messages are prioritized for translation. Whether that happens in Candida, we haven't looked at.
Great. Thanks very much. Well, I'm getting the information from Jules that we're coming right up on the half hour. So I want to close by thanking again our two illustrious speakers, Mike Lorenz and Claire Hoving, and my co-host Ilyan Ilyev, and Jules and the rest of the Exeter crew for all their work making MycoTalks such a spectacular series for our community. Thanks to everyone for being here and for all the questions and discussion that your talks provoked. We really appreciate it. Enjoy the rest of your days, and thanks again for being here.